This is a production of Cornell University.
So thank you all for being here today as I talk about this work that I've done so far in my master's. So as you can see by the title, I'll be focusing on my work of tracing nitrate transformations in forest soil. So a little bit of the background as to how I got started on this project. One of my advisors, Christy Goodale, started a nitrogen tracing experiment about 10 years ago in Arnott Forest, and they found some interesting results. And upon further discussions, we decided that doing a lab project kind of as a jump off from this work would be a pretty interesting avenue to go. So as to why this is of interest, temperate forests ecosystems are nitrogen sensitive, largely because they have been traditionally very nitrogen limited. And with the amount of human activity increasing the amount of reactive nitrogen going into the global nitrogen pool, including terrestrial ecosystems, there have been kind of a wide range of effects, some of which have been speculated to possibly be positive in terms of increased growth from fertilization effect that might help offset CO2 emissions. But a lot of the effects that have been observed have been more on the negative side for like decreased water quality from eutrophication due to increased algal blooms from, again, this increased amount of available nutrient, as well as soil acidification due to deposition in the form of nitric acid. And there have also been a lot of observed decreases in biodiversity from changes in plant dynamics and competitions. So to give you a broader perspective of some of these fluxes and pools, now here you can see the pre-anthropogenic sources of lightning fixation, bacteria fixation, which is how ecosystems primarily got their nitrogen before the greater inputs of human activities. And these were all, this nitrogen was pretty tightly cycled within organic matter, uptake in plants, and then mineralization kind of more in the transitory forms before being recycled again, or lost through microbial respiration. So with anthropogenic activities, we also get reactive nitrogen in the form of fossil fuel emissions and in the form of fertilizer inputs. So some of the numbers here, you can see atmospheric deposition. You can have wet deposition or dry deposition as ammonium or nitrate or sort of solid particles. And we did have some at the early stages of the Industrial Revolution, but it's really increased a lot since then. You can see like the, again, kind of pre-anthropogenic natural fixation has more or less remained the same, slight decreases, probably due to decreased need from these other inputs. Agricultural crop, you know, a much smaller portion, but still noticeable. And here you can see, like, with industrial production from fertilizer and fossil fuel emissions, we've more or less doubled the amount of reactive nitrogen going through ecosystems. So, again, to kind of tie it back, this can have some pretty significant effects on natural ecosystems. So we're really interested in what happens to this nitrogen once it does go into some of these. So I'll be focusing on wet nitrate deposition. And here you can see some of the other fluxes that take place once it goes into the soils. So you can get direct plant uptake from ammonium or nitrate or even as amino acids. And you can get a lot of microbial biomass processing, and they can convert between these different inorganic forms as well as removing nitrogen from the system altogether as gases and through losses or incorporation into soil solids. So kind of trying to give you a really brief background for redox theory, what it is and why I'm interested in it. So as I mentioned, microbes can respire this nitrogen in the soil, and the reason they would do that is primarily under oxygen limiting conditions. So normally they use oxygen as the terminal electron acceptor for respiration activities, but if they have water inundated conditions or even anaerobic microsites, they need to turn to other electron acceptors. And nitrate is one of their preferred ones. So further up the ladder equals more energy gained from using it as electron acceptor. So you can see nitrate is about as close to oxygen as you can get for the compounds commonly present in soils. You can see manganese is also fairly far up there, and iron, and those are also two elements of interest to us in this study. You can see, so going back to that mention of anaerobic microsites or water inundated conditions. When your soils are pretty dry, you have pretty conditions, plenty of oxygen for microbes to use for their respiration, and then for either mildly waterlogged conditions or only recently waterlogged, you get a slight oxygen loss, but not really severe yet. By the time you get to the really very oxygen depleted conditions, you mostly have your nitrate is mostly completely gone. So that'll be important later on for some of the reasoning of my experimental setup. So just kind of bring together the last two slides a little bit into a simplified diagram. Some of the processes that take place with nitrate and some of its dominant pathways, or you can get direct uptake into microbial biomass. Under oxidation conditions, microbial biomass also breaks down soil organic matter and releases nitrate or ammonium. And they can also uptake ammonium and reduce it or oxidize it to nitrate. And under suboxic conditions, nitrite is the first intermediary, and it's actually pretty reactive. It doesn't stick around very long, but that's because it is so reactive. So it can actually react abiotically with different compounds in the environment, such as soil organic matter. And it's also often reduced further and lost from the system. So kind of bringing this back to the reasoning for starting this project. So some of the things we know, so previous studies of nitrogen processing in soils have looked at the fates and processes on different timescales. So you have stuff that's taken place from minutes to days or weeks, and studies that typically focus around, what is the fate of it after 1 to 2 years after the initial application. But they typically don't really pair it well with investigating the role of the different soil horizons, as well as monitoring some of these other redox active species like iron and manganese. So we kind of wanted to try to get at that a little bit. So moving back to Christie's study, as far as the interesting results that got me questioning, thinking further, they did a seasonal nitrate application with nitrogen 15 tracer. So they applied this nitrogen 15 nitrate in spring, summer, and fall, and got some somewhat unexpected results, particularly for the spring application. So they actually had pretty high soil saturation in the spring with pretty low microbial respiration, which is often considered to be one of the dominant things for nitrogen added into soil system. So since they didn't appear to have much microbial activity, they would have expected more of it to go through kind of unaffected and into stream water as nitrate, or possibly to get respired and lost in gases. What they found was that they actually had really good retention, and speculated that perhaps this was due to some of those other abiotic factors, such as activity of other redox active metals that can couple with soil organic matter. And then in the summer, they had quite low nitrogen retention, which one of the other sinks that is presumed to be predominant is plant uptake. And so you expect lower plant uptake in the spring before plants have lifed out, and higher uptake in the summer when they're at peak growth. And they really didn't see much change from that. So yeah. So again, more questions as to exactly what is going on in these soils with regards to why and when nitrogen is retained. So from this, I had two main questions. One, how does redox status affect the fate of nitrogen in these in a forest soil? And how is the partitioning affected by horizon? So what effect does mineralogy and organic matter have? So to do this, to be able to do this, we wanted to follow partitioning of nitrate between the different soil and solution pools so that we could explore the impact of redox status and soil horizon on the fate of nitrogen. So for the redox condition hypotheses, we believe that we hypothesize that we would have greater overall system losses under suboxic conditions, just because we expect that microbial activity will definitely contribute to this, to gaseous losses. Under oxic conditions, we expect that higher organic matter translates to more microbial processing and more nitrogen partitioning with soil organic matter and dissolved organic matter. And kind of conversely, suboxic conditions will have with low organic matter, which will have presumably less microbial processing as well, will retain more nitrogen and soil organic matter and dissolved organic matter. We thought that all horizons will have incorporation to microbial biomass and some stabilization, and that low organic matter soils will retain more of the nitrogen unprocessed in the form of nitrate. So for our experimental site, as I mentioned, these are soils from Arnott Forest nearby, so a little bit southwest of here. We collect these soils from near the top of a hill slope, so they're pretty well drained. And they're also fairly shallow. We were only able to dig down to around 60 centimeters before hitting rocks that we couldn't go through. And these hardwoods have been, this growth has been established more or less since early 1900s, since some major fires in 1900, 1912. So we have, you can see here, we have a pretty well developed organic horizon, which isn't true for all of the soils in this area. A lot of them have had a lot of earthworm activity where there's not necessarily much of an organic horizon apparent, so we were kind of lucky in our choice here. These soils are also quite acidic, so it ranges from 3.5 to pH of 4. They're all silty loams or silty clay loams. And you can see, again, like it's pretty apparent there that there's a lot of carbon, but it's borne out in the numbers of 40% carbon in the O horizon and 25% in what we're calling the A horizon. That's sort of a temporary name right now until we kind of do some mineralogy to see if it should be A or O horizon, classified as mineral or organic horizon. And then for some of the chemical characteristics, you can see the O horizon has pretty high base saturation, and A is pretty high as well, but the O and the E quite low, which you can also kind of see verified by the high amounts of aluminum and iron in the E and the B horizons. We also have a relatively high amount of manganese in the O horizon compared to the others, which also could be of interest since it is a redox active metal. So putting the nitrogen deposition for the site into perspective, you can see here in 2000, total deposition was a lot higher than it is now. So still elevated above presumed pre-industrial levels, but deposition is moving in the right direction. And you can see for nitrate in particular, makes up a pretty good proportion of the overall deposition. Yeah. So as for the experiments themselves, I took these soils, separated them into the different horizons, and made them into slurries in a jar. So I had 1:8 soil to solution ratio. And for the different conditions, I bubbled in oxygen or nitrogen for the suboxic experiments that are actually yet to come. I haven't gotten to do those yet. So all the results I will present are for oxygen conditions only. So I wanted to chase, so I spiked in my nitrogen 15 label, and I wanted to trace them through time, so kind of on the order of a one month time. So I periodically took samples and centrifuged them down to separate out the soil and solution portions. And I also did the same with a control that did not have any labeled nitrogen. Since I am interested in the partitioning into specific pools, I did a whole range of analytes on the solution and the solids. So both for total quantities as well as for isotopic enrichment. So isotopic enrichment in the soil solids as well, and microbial biomass. So as I mentioned earlier, you kind of see the region in which nitrate is active is really predominantly in the suboxic conditions. So we really didn't want to go into anoxic because then we wouldn't really have much, it would be much harder to trace it on any kind of time sequence. We would expect it to more or less disappear immediately, and we didn't. We wanted to kind of drag it out a little bit. So some of the chemical species that are active in these regions, as I mentioned before, monitoring metals as well as these different forms of nitrogen. So where Anid mentioned that electronics, this is where that comes in. So I wanted to measure my redox status with an ORP probe, so oxidation reduction potential probe, as well as monitoring by these chemical species. But we didn't have a meter that was capable of running 6 probes at once. And if I tried to use all the meters that we had in the lab, I would basically take them all up and take up too much space. So I did some digging and I realized that I could make my own meter. And so I did. So I used an Arduino as well as some parts from Atlas Scientific here to assemble my own meter, combination data logger. So I wrote the code for these things and was able to get some nice time resolved data. So I didn't have to, go in 3 times a day and manually write it down, lose more resolution. So this was a fun little side project. As I mentioned, with this handy dandy little meter, I monitored for redox potential and pH during the first 3 days of incubation. The reason it was only the first 3 days was partially that I didn't want to contaminate my probes with nitrogen-15 and potentially skew my future results. So that's also where the use of the chemical indicators comes in as a way to monitor for redox status after we pull out the probes. Just a brief overview of the methods I did for this whole suite of analytes. Did a lot of colorimetric reagents, metric analyses. The Shimadzu autoanalyzer for dissolved organic carbon, total nitrogen, atomic absorption for total manganese in solution, chloroform extractions for microbial biomass, and combustion mass spec for total carbon and nitrogen in soils. And then for the nitrogen-15 tracer, submitted samples from all these different potential nitrogen pools to coil. Microdiffusion onto acidified disk was particularly fun. I don't know if any of you have run that method, but it is particularly tedious and time consuming. Yeah. So moving into the results, the next few slides I'm going to show are kind of for the total analytes in solution. On all of them, I have dotted lines indicating my detection limits for the various analytes. And blue arrows indicating when the spike was added. In this slide, the yellow diamonds also indicate the quantity of nitrate added. So you can see the amount of spike that we added was calibrated to try to get a quantifiable increase in enrichment, but not so much that we really change the native nitrogen dynamics. So we didn't want to add 5 ppms of nitrate nitrogen. So you can see there seems to be a little bit of total nitrate concentration change, but it's not crazy. And again, everything I'm showing you now is for the oxic conditions. So we really didn't see any nitrate at any given time not surprising. Would expect that more in the suboxic conditions. The O horizon was the only one where we really saw the complete disappearance of the nitrate. The other horizons, the nitrate more or less remained steady throughout the experiment. We did see a lot of ammonium evolve during the course of the 30 days. And so here I'm showing ammonium ammonium in purple and total nitrogen in green. We expected to see a larger difference here. Usually there's a lot more organic nitrogen evolved. So we're investigating right now why we're not seeing that. Potentially an underestimation from some of our analyses. Possibly real, but it would be surprising if it was entirely absent in, say, the A horizon. So again, we see an increase in dissolved organic carbon as well. Also kind of expected you'd expect that with microbial processes, release of nitrogen in the soil, as well as abiotic release of more labile carbon and nitrogen. Metals remain pretty stable. Again, we don't really expect to see too much dissolution of metals under the oxic conditions. The only one that was like particularly surprising was this increase in soluble manganese here in the B horizon. It might help explain one of the results I'll show next, but I'll come back to that in a minute. So now kind of the meat of the project. Where does the label go? So we found that it does primarily go in, that we do get a lot more incorporation to the solids in the higher organic matter horizons. So highest with highest organic matter O, next with still high organic matter A. The E horizon was a little surprising in that it was lower than the B, and also was the only one that didn't seem to have reached kind of stability in the same time frame. The others, they've more or less leveled off by day 15, but E horizon is still going. So it'd be interesting to see, like, if we had continued it to 60 days, what level it might have stabilized at. Here again, so purple is the soil nitrogen recovery that I showed in this plot here. And you can see, like, one of the bigger takeaways is that you can see in the E and B horizons, a lot of this nitrogen is still in the form of nitrate. It never got processed. So the B horizon is kind of interesting in that it had the lowest total recovery of all of them, which, going back to that manganese, might be an indication that we had more reducing conditions than we thought we did, than we tried to control for. Oh, forgot about microbial biomass. So the measurements for the microbial biomass were a little difficult, and we got a lot lower recoveries than we had expected, which makes quantifying the amount of nitrogen tracer recovered in there a lot more difficult. But from the calculations we have done, it seems to be a pretty small proportion of it for any given time. So moving back to the hypotheses, two of these we weren't able to address at all at this point because we haven't done the suboxic conditions. Those will be continued throughout the summer. But we did find for oxic conditions, higher soil organic matter did correspond with more nitrogen partitioned into solids, presumably into soil organic matter. Again, we weren't really able to dive too deep into microbial processing or dissolved organic matter, so we would have to improve our techniques to get better quantities of those. Let's see. We also did find that all horizons did have stabilization into soil solids, and they all did have some release of ammonium. Again, DUM, a little problematic. And we also did indeed find that lower soil organic horizons had more unprocessed nitrate. So kind of trying to take it back to my original questions of effects of soil horizon and redox effects. Again, not too much we can do with the redox effects at this point, but we do clearly see that horizon soil organic matter has a very strong effect. And again, until we get into the suboxic, it's kind of hard to say what effect the iron and manganese minerals might have, because we really don't see a whole lot of activity there under the oxic conditions. With that, I'd like to thank the many people who have helped me along the way, my advisors, Anid and Christy, as well as lab mates past and present who braved the wild ticks and unexpected rainstorms to help me make this possible. And thank you all.
I was wondering, are you thinking you guys in the manganese storage? You have an ability to form different oxidation states of manganese in your solution. And manganese seems to be a hot topic. Everybody jumps on manganese at the moment. Seems in this context that not many people look at that.
Yeah, I haven't seen many people in relation to nitrogen in these systems. Yeah, manganese. But I don't know of any way to probe oxidation state. We'd love to. I don't know. Have you come across any? Okay. Yeah. So far, we haven't found any. It's just total manganese in solution.
I'd follow that up by asking, do you have an idea about where that manganese is coming from? Why is it so high? Or is that high?
Yeah, I don't really know if it's high in a broader sense. In the soil properties table I showed you, manganese in the B horizon was a lot lower than, say, in the A horizon or the O horizon. I should actually look into what these levels of manganese are in relation to other soils.
I found it interesting that the levels were actually, I mean, maybe I was reading this wrong, but they seemed like they were higher than iron.
Yeah, higher than dissolved iron.
Which is really surprising.
Yeah, I think that's partially due to it being higher on the redox ladder. You know, people talk about iron reduction in soils a lot, but I'm not sure how much that is because it's just so much more visible and present in the soils in higher quantities that manganese mostly gets overlooked. Yeah, I don't know how frequently people measure dissolved manganese.
Well, the oxidized, I mean, just by looking at the oxidized form, that's where the color, that's why people talk about it that way. But if you're talking about the reduced form, there's not a lot of, visually you can't identify reduced manganese, but you can with reduced iron. So but again, I don't know what that ratio is to iron. I mean, the reason why iron is talked a lot about it, other than its visual, is that it is ubiquitous and in quantities that are very much higher than manganese in the terrestrial system in general. So it's interesting that you found that there, but it's related to respiration.
could any of this still be related to the fires? Because I was thinking that surface layer, you mentioned there were 2 major fires, I know that was some time ago, but if there's any mineral matter that was up in the trees, any of the ash will still be in that O-horizon unless it's runoff and not really a high runoff spot. I wonder if they're looking to the B horizon and the O horizon. Wondering if some of that is fertilized or supplemented by the fact that I don't know, I didn't realize about how severe those fires were, I don't know.
Yeah, I think they were pretty major. We have actually found some paralyzed carbon that we speculated is still remnant from those fires. So we get some fairly large portions of what looks to be paralyzed material.
Thanks.
Okay, I haven't thought about the ash. But yeah, sure.
So I'm not a soil chemist, but our soil health work, manganese came up many times as an important indicator, which surprised us. And then somebody pointed out to me that the redox reactions are also very instrumental in the decomposition of organic matter. So it's not only the relative availability of oxygen, but it's also the process itself of decomposition. Does it make any sense? But I can send you a paper. You probably understand this a lot better, but somebody pointed it out to me that's one of the reasons why manganese 2+ is then often more available. Does it make sense? Or?
Just because of its biological importance?
Yes.
Okay.
That as part of the decomposition process, that is a reduction when you go to 2+, or is it
4 to 2.
4 to 2 is what? Reduction?
Reduction.
Yeah. So that it's a reduction. I'll send you the paper.
Okay, yeah.
But it does kind of explain that you have much higher levels in the upper horizons. For what it's worth.
Are you going to publish your method for making those probes in any way, shape, or form? Because I've thought about that kind of stuff too. It's really handy to have that.
Yeah, I do want to publish it on GitHub.
Okay. There's Jove as well, Journal of Digital. Oh, maybe I have to take pictures. There's a journal dedicated for methods called Jove. I think it's the V stands for visual, so I don't know. You meet those criteria, just take pictures. Make a GIF.
Okay.
Thank you again.
This has been a production of Cornell University, on the web at cornell.edu.